Hello, I'm James Andrews, reporter of Food Safety News. I'm here at IAFP 2015. It's a food safety conference in Portland, Oregon. And joining me is Dr. Lawrence Goodridge. He's a food safety professor at McGill University. And Lawrence, I have you here because your research team was recently awarded a really big research grant to study Salmonella. And when we got the we get lots of press releases at Food Safety News about all sorts of research, and when we saw yours, our eyes kind of widened at the size of the grant, the $10 million grant to study Salmonella, because I don't think we've seen a research project of that size specifically for Salmonella. I know you were talking about recent USDA grants to study Shiga toxin producing E. coli in recent years for $25 million, and another one for neurovirus for $25 million. But Salmonella specifically, you don't see research projects of that size. Can you tell me how that came about, how you guys got that, and a little bit about what the initial idea for what you wanted to do was?
So the initial idea came from the fact that we had previously conducted environmental sampling in agricultural environments where fresh produce was being grown, tomatoes, cantaloupes, things of that sort. And we saw that we routinely isolated different Salmonella serotypes, and when we determined that those serotypes, they seemed to be different than the ones that cause human disease. And so we were wondering about that, because we typically see 20 to 25 serotypes or types of Salmonella that cause human disease, and yet there's more than 2,500 of these different types that have been identified. So we were questioning, well, why is it only 25 types that cause the majority of human disease, and what is the nature of these other 2,500 plus types that are in the environment but don't seem to make people sick?
Right, and why Salmonella? I mean, Salmonella causes a ton of illnesses in North America and worldwide, as we were talking about earlier. But what do you think are still the remaining big challenges that we're facing related to Salmonella?
The first thing is Salmonella causes approximately 93 million cases of foodborne illness worldwide each year. In North America, it's one of the leading causes of illnesses, hospitalizations, and deaths. And we still have so many questions to ask and answer about Salmonella. For example, Salmonella can cause outbreaks in a plethora of foods, including dry foods, which raises questions about their ability to survive in places where we don't normally see other foodborne pathogens surviving. With respect to fresh produce, fresh fruits and vegetables, Salmonella now accounts for almost half of the outbreaks that are caused by those commodities, and there are many questions regarding why that is. So in choosing to study Salmonella, our hope is to begin to answer some of the many questions that remain with respect to why this pathogen causes so many illnesses worldwide.
How are you going to be going about doing this? I understand the first phase of the study is going to be a big genetic sequencing project. Can you talk about step one and then how that's going to lead to step two, step three?
Sure. So as I said previously, there's 2,500 plus types of Salmonella. Our goal will be to sequence 4,500 of those isolates from those different types to obtain information regarding the DNA sequence of the whole genome of the different bacteria. And those bacteria will come from the types of Salmonella that we commonly see causing illness in humans, as well as types of Salmonella that are rare or perhaps have never caused illness in humans. And we hope to compare the genomes of those different types, and when combined with other methods, obtain information that will tell us more precisely why there are certain types of Salmonella that seem to commonly cause illness, and why there are other types that do not seem to commonly cause illness in humans.
Right, like say this virulent Salmonella serotype has this, all these virulent, this like dozen virulent types over here have this certain trait that you don't see in, say, another 1,000 serotypes over here.
Right, and some of that work has already been accomplished in many studies, but certainly not in as comprehensive a manner as what we hope to do in this work.
And are you going to be using newer technology than perhaps some of those other studies?
Yeah, so that's exactly. So that's another reason for doing this work. We will be sequencing the entire genomes of the bacteria that we're testing, which again has been done previously, but on a small scale with a small number of isolates. In our case, we will be sequencing 4,500 isolates of Salmonella obtained from all over the world. And again, while most studies have focused on only the ones that seem to cause human illness, we will expand that and focus not only on those types, but types that are rare, in order to really try to understand why it is that there's only a small number of Salmonella that seems to cause the majority of human illness.
How are you going to end up using this information? I mean, what ways might this information help companies or help researchers prevent Salmonella contamination?
So the objective of this study is to ultimately reduce the number of Salmonella cases that occur through consumption of contaminated food. And so this information will help us to do that in three ways. First, we'll make use of the information to develop novel strategies to control Salmonella on fresh produce as it is growing in the field, so that is pre-harvest. Secondly, the information will be used to develop better and more accurate diagnostic tests. So for example, if we can show that there are some Salmonella that are more virulent than other Salmonella, and we identify the markers in the bacteria that cause one Salmonella to be more virulent than the other, then we can actually build that into the new test, so that when we detect Salmonella, it will not only tell us that Salmonella is present, but it will tell us about its ability to cause human disease. And then finally, we hope to develop new methods that could be used to track the sources of contaminated food during an outbreak, so that those foods can be removed from circulation from retail or grocery stores more faster than is what is currently happening.
Okay. And the last question I wanted to ask you is, so how far have you gotten? I know you're just at the beginning of this study. What are the next 4 years going to look like? Lots of time in the lab. Just what's your status right now, and how are you going to move forward?
So the project will not officially begin until October. However, we've already began to do some preliminary work. Certainly, the next 4 years will be a lot of time spent in the laboratory, but it's also important that we disseminate the information to the government, to industry, and to the general public, so that they are kept abreast of our progress in this project.
All right, well, Dr. Goodridge, I really appreciate you taking some time to talk with us about this, and good luck with everything.
Thank you very much.
We'll probably be covering it.
Thank you.